Thanks,
Is that
buddy.
better?
It's recording, but we still got the boxes.
All right, so I have to go into share again. Looks like. Oh my God. New share. Still the same?
Yep.
Of course it is. Okay, I see all you guys. This can't be this difficult. It really can't. All right, let's try this. Let's try
Well, now I can see part of your desktop.
that. Yeah, nothing interesting, I'm sure.
No.
That didn't help?
No.
This doesn't make any sense.
Hey, I'm not Isaac anymore. I'm Bob.
Oh, you're Bob? All right. Okay. New share. Okay, basic. You gotta be kidding me.
There we go. Yeah. Yes. Oh, yeah. Yeah, like, how could it be contracted and how can it cause possible, maybe temporary dementia? I don't know. Yeah, he went in for a cortisone shot in his back or his neck, wherever they did it. But it's not his first one. You know, he's done it before. So when he did that, he had like this just crazy, immense pain. And it kept getting worse and worse, and he wasn't sleeping. He was up all night. And finally, he went to the hospital on Friday, and they did a whole bunch of blood work, and that's how they found the osteomyelitis. But also, like mentally, he's not not all there, and he's usually, you know, like a bright guy. He has Parkinson's as a like a, you know, whatever part of his medical history. After the blood work yesterday, but they're doing a bone biopsy tomorrow in his back. Yeah. 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 Well, they're running tests and stuff, so I don't know. We'll see. I'll let you know.
Yeah, I had a question. Yeah, so can you just explain how it works? So say you had the E. coli and you didn't use the mordant. It's gram negative. It's thinner than the other. Yep. Mm-hmm. 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 And then you do the other dye. Was it safranine? Safranine? Oh, you do alcohol. Yep. Okay. But the question was that if you didn't use the mordant, what color would it be? So it would basically be, it would affect it the same way, basically. If you use the mordant and then applied the alcohol, it removed that purple color. If you didn't use the mordant and you applied the alcohol, it would remove that purple color.
Yes.
Okay, all right.
Yeah. So what was the answer? Was still pink, or what was the question?
No, yes, it was still pink because I was thinking it would be purple, but it makes sense. If it's either way, it doesn't, the mordant really kind of, you'd still end up with pink.
Well, yeah, you're gonna use the crystal violet and use the mordant, which is a salt. So now you have one molecule of the crystal violet. You add the salt. Now that becomes a compound, right? It goes in as one. You add the mordant. They lock together.
Mm-hmm.
So it's two. So it's much harder to come out because it's bigger than when it went in.
Okay.
Yeah. So if you don't add the mordant, they're really not going to lock in. Some of the purple will probably stay in the gram positive, but they're not going to really lock in. All right. And then you're going to still use the decolorizer, right?
Mm-hmm.
The decolorizer is going to get most of the coloring out of, well, it's definitely going to get all the coloring out of the gram negative.
Okay. Okay.
Most of it out of the gram positive. Then when you counterstain, the gram negative will usually always be pink because you've removed all the crystal violet because it's so thin.
Okay. All right. Okay, that makes more sense.
That was a difficult question. You got that one.
Yeah. I was just like, I was just going, wait a second. I wasn't quite sure how that mordant really worked, I guess. But okay, it makes more sense.
I can fix that. Don't worry.
Okay.
I will fix that for you.
Oh, okay.
But that's a good question. That was when I was putting it in, I'm like, oh, that's nasty.
I was trying very hard to figure it out, but yeah, I didn't figure it right. Okay.
I think there was a chart in your book, and I remember I asked you guys, I would ask.
No, I remember talking about it, but I didn't think about like not having the mordant, how that would affect it one way or the other.
Yeah.
I didn't picture that in there.
If we were in lecture and lab, I'd be drawing on the board, and then I'd ask you guys that exact question.
Yeah.
Yeah, you guys are at quite a disadvantage. All right. So did I skip chapter 19? Am I losing my mind?
I feel like you mentioned chapter 19,
Yeah,
but I
but
don't
did we go
remember.
over it? This doesn't look familiar. I thought we went over.
What is it?
What was in chapter 19?
Yeah, what's in it?
It's going to be, as soon as I get you guys out of the way here.
Because I'll be honest, I'm not keeping track of the chapters. I'm just...
Yeah, they should follow a numerical order. So this is chapter 19. I don't think we went over this.
Checking my notes.
Can you go to like slide 6, the picture? Just maybe the picture will prompt me. This doesn't. No, I don't think we went over anthrax.
All right, so I'll go over 19. No. Were you guys able to the Zoom videos upload into Blackboard?
I think I saw them in there.
Yeah, because those I'm converting them to my YouTube channel, and then from my YouTube channel, I'm embedding them into Blackboard. That's the only way they're working.
What's your YouTube channel?
Never mind, Bob. I don't know. It's like, it's Dr. Philip Edward Jones, but I think it might be under, I think that's what it is.
All right.
Pretty sure. But I'll upload a link. So I want to take everything from ECC, our biology page that are mine, and upload them into my channel, and then I can upload them into Blackboard for you guys.
I have another question. So I took Unit 2 test, but was it supposed to be worth 500 points? Because each one was 10 points. So I ended up, out of 500 points. So, I mean, it's basically the same thing if you just
Yeah, well, I have to go.
divide it out. But
I'll just convert it.
okay, all right, yeah.
All right, yeah, thank you because on my end, when I hit, it says 50 questions, 2 points is 100 points. And then when I upload it, I don't know what Blackboard does to it.
Yeah, okay.
Yeah, it's fixable.
Okay.
All right, so let's get started. I'm gonna make sure nobody else wants to get in. Okay, so there's like 6 of us. Okay, great. All right, so we probably should be able to wrap up Unit 4 today or tomorrow, and then we only have the last unit to go. So I think we're in really good shape. And like I said, I'm going to load up all the tests, and you guys can do it. The course ends on the 12th, but we have like 4 or 5 days to play with until the grades are due. And Blackboard's going to tabulate all those grades for me anyway. I just have to go through and take them off Blackboard and put them in the WebEx. So don't panic if we're running out of time or things come up, okay? All right, can everyone see that?
Yep.
Okay. So we're going to do medically important Gram positive bacilli. So what can you guys tell me about Gram positive bacilli right now without looking?
There's a thick cell wall.
The glycan layer, yeah.
And then it has just one membrane, cell membrane.
Okay, and then how would we, if we were thinking about getting rid of Gram positive bacteria, what would we use, do you think? How would we effectively get to that?
Something that deals with the cell wall, like penicillin?
Yeah, penicillin. So we want to get something that's going to break up those NAN and NAG units. Go in. Same as the lysosome in your tears. They basically do the same type of thing, all right? So these can be divided into 3 groups based on the presence or absence of endospores and acid fastness. So if I asked you guys something about what was more virulent or what would be a virulence factor, can you guys tell me anything about endospores or acid fastness that we talked about so far? I know it's a lot. I'm just trying to get you thinking. Right, what do we know about endospores?
Very, very, very hard to kill.
Yeah, so we can only really kill them autoclaving, ethylene gas, and peroxide will do it with a high enough concentration, long enough duration, and remember we can do that tyndallization method too. But they're basically extremely hard to kill. We're not going to kill them randomly, right? So they're pretty virulent. All right, so non-endospore form is going to be a little bit easier to kill. All right, what about acid fastness? I know I mentioned it in chapter 3, but what do you think that means if it's acid fast?
It has a high pH.
Well, it's not about high pH. Usually they have like a waxy coating that we can't really
like TB.
get. Yeah, so they're going to have like a thicker coating. So even if we were able to phagocytose them, we'd have difficulty killing them. And we're going to talk about TB today and just realize TB, I know this is recording and someone's going to probably tell me I don't know what I'm talking about, but it's really not that easy to catch. Remember, you have to have constant exposure and you have to have enough of infectious dose, you have to have enough of it. And usually your body, if a small enough dose, it will kill it off. It's just when your body gets overwhelmed because they're so hard to get rid of that your body will sequester those. All right, we'll send a bunch of monocytes around it to surround it, and then you can get like a Gon tubercle, but we'll talk about that. All right, so gram-positive rods, we have our spore formers. So any of these, so when you guys, in your next exam or your board exams, if you see B something or C something, it's abbreviation for genus, for Bacillus or Clostridium. So whenever you see those, I want you to automatically think it's an endospore former. All right, so the vegetative forms are going to be easier to kill. And what do these do when things get dangerous or when nutrients are not available or things look not so good? What are these going to do?
Kill the host.
Oh, they're going to actually form spores. Remember, they're vegetative cells. When things get when the water's low or nutrients are low or something's in the environment, they literally go through that. They divide, binary fission, right? And then the one wraps around the other and they make that calcium oxalate coating around them. And then they become spores. Remember, these spores can be around for tens of thousands to a million years, all right? And they can literally come back to life if their conditions get much better, right? So non-spore formers are easier to kill, all right? But we're gonna find out, you guys heard of listeria?
Mhm.
Okay, so listeria likes to grow on lunch meat. It's an intestinal bacteria. It's usually on lunch meat or processed food. And this is going to be one of those that tricks your monocytes into engulfing them. All right, remember the advantage of that is they go undetected in your body because your body, when your immune system sees a monocyte, it sees it itself. It doesn't attack it. So these things literally multiply in the monocytes, and they can leave whenever they want, right? And then mycobacterium is acid fast, so it's very, very hard to stain. It's got that very, very almost a waxy coating, so it's also very hard to kill through phagocytosis, right? And Corynebacterium and Propionibacterium, we're going to talk a little bit about that. It causes some major throat issues in children with pertussis. All right, it causes that, a pseudomembrane in the back of the throat, and Propionibacterium is usually the leading cause of acne. All right, so that'll be a question that you'll probably see coming up at some point. So remember, Bacillus and Clostridium, these are going to be spore formers. So there's a spore right there. There's a vegetative cell. So these are vegetative cells. If things are good, they just keep reproducing. All right, but when things get a little rough, they start forming spores. All right, and that's a safety mechanism for them. Right, there's another spore. Right. So these are spore formers. Just kind of keep that in the back of your mind. All right, so these are gram-positive endospore forming, anything bacillus. So it's going to be B, and then Bacillus cereus, or these other ones are going to, remember, they're spore formers. There are motile rods. They're mostly saprophytic. Does anybody remember what that term means?
Like fungus will eat dead
Yeah.
things.
Yeah, so these are going to be bacteria in the soil.
Mm-hmm.
Right, they're in the soil all over. They're ubiquitous, and they go in and they just decompose things. So we kind of really want them on, right? They're aerobic and catalase positive. Right? So they have a way of dealing with oxygen. Versatile and degrading complex macromolecules. They just break things down, and we need them in the soil, right? Source of antibiotics, right? So these are going to they're going to release enzymes in their environment that will kill off everything around it. So it kills all of the competition, and that's how we use that enzyme to derive antibiotics. Right? Primary habit in the soil. Two species that we're going to talk about are Bacillus anthracis. All right, so you guys have all heard of anthrax.
Mm-hmm.
Right, it's in the soil, right, and it's usually, usually it's in soil. And Bacillus cereus, all right, this can cause major food poisoning, right? It's food intoxication. Right? So Bacillus anthracis, they're large block-shaped rods, so they're large. Can't miss these. Central spores that develop under all conditions except in the living body. Right? So we're going to find out they're usually cutaneous. Right? So they're usually on the surface of your skin. Virulence factor, they have a polypeptide capsule and exotoxins. So exotoxins are going to secrete these toxins. And what do you think about this polypeptide capsule? What do you think how is that a virulence factor? All right, so peptide, what are you thinking immediately?
Proteins.
Protein. Poly means many. So it's going to be a very thick, proteinaceous capsule. It's going to be very hard to get through if we want to kill this, okay? So there's three types, right? So this is anthrax. Cutaneous, spores enter through skin, all right? Whenever you see the word eschar, that usually means black. We'll see it again at the end of the semester. This is the least dangerous, right? Now, what do you think about pulmonary?
That's no good.
No good, right? Because didn't somebody try to put
anthrax in the mail recently, a while back, yeah.
People can't see it, they inhale it, they don't know they have it until it's, if they have an infection. All right. And then gastrointestinal. So we're ingesting the spores. And remember, they're going to be have a protein coating on them, and they're going to start releasing exotoxins into the environment and cause major inflammation.
Yeah, try and get them covered.
Can somebody mute whoever's? Sorry. All right. So we're going to treat it with penicillin, tetracycline, or ciprofloxacin. All right. And just realize there's vaccines, live spores, and toxins to protect livestock. Right. Purified toxoid for high-risk occupations and military personnel. So we're just saying we're not going to vaccinate the general population for in most conditions, but if you're in a high-risk occupation, so if you're on the front lines or you're in an area where it's relatively high concentration of soil or you're breathing it in, we're going to vaccinate you for it. Military personnel, definitely. It's another one that we have to give boosters to. All right, even though we have memory cells to it, we want to boost that periodically for them. Okay. Now, Bacillus cereus or B cereus is common airborne and dustborne. Right. So, usual methods of infection and antisepsis are ineffective. Right, it's airborne and dustborne. So, we can't even clean a surface. We're going to be breathing this in. Right, it can cause gastrointestinal distress if we consume it. Grows in food, spores survive cooking and reheating. All right, because we're not autoclaving this. Unless you're using an Instapot, you're not going to kill these spores. So what do you think happens to the spores once they're in your GI tract? They've gotten past the acid of your stomach. Now you're in the duodenum and your small intestine, all right? And we know the soil isn't always full of oxygen. So these things can actually survive in the lower oxygen concentrations. And if they're spores, we're going to say, oh, things are great. I'm going to just turn into vegetative cells and they're going to flourish. They're going to start releasing those toxins, exotoxins, causing nausea, vomiting, abdominal cramps, and diarrhea for about a 24-hour duration. So if you're in good health, you're going to be, your stomach's going to be upset. You're probably going to have a little diarrhea. You're going to flush that out in most cases. Right? But here, once again, remember a lot of these things increasingly reported in immune suppressed because these people have other things going on. So things that wouldn't be quite as pathological or as aggressive in us as healthy individuals are going to be going to make more of a difference in the immunosuppressed people. Okay. Now we're going to talk about Clostridium. So if I said C. diff, would everyone know what I'm talking about? Mm-hmm. All right. So Clostridium difficile, right. Gram-positive spore-forming rods, anaerobic and catalase negative. All right. So they can't really deal with oxygen as well. So where are they going to live if they're living in your GI tract? They're going to live more distant.
For the colon.
Deep in there, yeah.
Yeah, they're going to live, the large intestine because the oxygen concentration there is going to be the least. Ovular spheroid spores produce only under anaerobic conditions, and your large intestine is relatively anaerobic. These synthesize organic acids, alcohol, and exotoxins, so that doesn't sound like it's very good. So this is why we don't want these overrunning our GI tract. And once again, if we remember, if we go through an aggressive antimicrobial therapy, you're wiping out everything but the spore formers. So the only thing that's going to be left is C. diff in your large intestine. Please realize most of us have some C. diff in our intestinal tract because we've consumed it through dirt or whatever. We've consumed it. It's in your large intestine, but we're keeping it at bay there because there's other microbes there for competition. It's when we wipe out everything else, C. diff's like, well, I'm the only one around. I can grow as aggressively as I want, and I can synthesize organic acids, alcohol, and exotoxins. Right? Can also cause wound infections, tissue infections, and food intoxications if it's consumed. Once again, these spores are going to start releasing, or this C. diff is going to start releasing those organic acids, alcohol, exotoxins in your GI tract. If it's in your tissues, if it's deep enough, which anaerobic, it can start thriving in there, in your wounds or tissue infections. We hope we never see it. All right, but Clostridium perfringens, all right, this is the component that causes gas gangrene. Most frequent clostridial involvement in soft tissue wound infections. So myo, whenever you see myo means muscle, necrosis means
Death.
dead. All right, so this stuff literally will get deep in your tissues. It can't live superficially. It's got to be deep where the oxygen concentration is low. It starts multiplying in there, and it literally starts consuming your dead tissue from your wound. All right, it lives off the carbohydrates in your wound. So with gas gangrene, you have to clean that wound out aggressively. And then what we can do is we can start hitting it with oxygen, because remember, this can't really deal with oxygen, right? Spores are found in soil, human skin, intestines, and the vagina. Predisposing factors: surgical incisions, compound fractures, diabetic ulcers, septic abortions, puncture wounds, and gunshot wounds. Now think about that whole litany of things right there. What do you think those have in common?
Typical night at ECMC.
Yeah, right. Okay, well, they're deep, all right? They're not, these are not, none of these are superficial. Surgical incisions, you're going in deep. You're breaching that first layer of skin and everything. Compound fractures, all right? So that's when the bone literally breaks and punctures the skin. So if it gets in deep enough into that wound, diabetic ulcers, right? If you've never seen these or known what these are, so people that are diabetic, I mentioned last class, they lose their entire capillary bed, remember? And all of their tissue starts dying, all right? And these can get deep into the tissue where there's no oxygen, they can grow. Septic abortions, all right, you know, we don't have to go into how that is, but abortion where these were implanted deep into the reproductive tract and they're deep in the tissue, they can survive there, right? Puncture wounds, you're puncturing into the wound, you're going deep, you're actually implanting these, impregnating these spores deep into the skin. And gunshot wounds, think about that. We're shooting a gun, a gunshot wound is going through your skin deep into your tissue. So if there's any Clostridium on that, it's deep in the tissues, it can start surviving. All right, so I'm going to mention this quickly. So when you get into the healthcare field or whatever, and someone has diabetes, all right, and you want to check their feet out, make sure you take their socks off. Because if you're just palpating their feet and you can't see that they're gangrenous, when you go to manipulate the toes, they'll snap right off. Happened to a friend of mine. I said, "Oh my God, did you shit yourself?" He's like, "Yeah, it was pretty bad."
Oh my gosh.
So yeah, can you imagine like having a little Tootsie Roll come right off it looks like a Tootsie Roll.
And the smell is awful.
Yeah, well, it's all rotting tissue.
Yeah.
Oh my God, is everyone seeing that?
Oh,
Running
in time. Yep.
I can't believe this. Okay, I'll just restart again. Oh my God. All right. So variance factors. Toxins, alpha toxin causes red blood cell rupture, edema, and tissue destruction. So this, once it's implanted, it's growing, it's releasing alpha toxin. It's literally destroying all the tissue around it, especially with gas gangrene. It's going in, it starts, the toxins start eating away at the tissue, killing it. Now the Clostridium has all kinds of carbohydrates to live off of your dead tissue. Collagenase, you guys remember, that'll eat through any connective tissue. It'll just eat its way through wherever. Hyaluronidase, a component of cartilage, starts digesting that. And anything that contains DNA, it'll just start digesting it. So it's got some major virulence factors. So the pathology of it, not highly invasive, requires damaged or dead tissue and anaerobic conditions. Right? And that literally, in case you can't tell there, that is someone's left thigh posterior. Pretty nasty. Conditions stimulate spore germination, vegetative growth, and release exotoxins and other virulence factors. So these get in, conditions are, they can start going through binary fission and they start releasing exotoxins. They just eat their way through wherever they are. Fermentation of muscle carbohydrates results in the formation of gas and further tissue destruction. So it's called gas gangrene because it's fermenting carbohydrates. When it's fermenting, it's giving off gas. Right? You guys are, you're not going to see that in the biological test and lab. And we really don't keep this around, but it starts fermenting, and as it fermenting, it starts forming gas, and it literally makes pockets in the tissue. So treatment, I said we have to really aggressively clean these out. Immediate cleaning of dirty wounds, deep wounds, compound fractures, and infected incisions. Right? Debridements, we have to scrape or cut off all that dead tissue. And sometimes you will have to cut off some healthy tissue too, just to make sure you get it all. Large doses of cephalosporin and penicillin, hyperbaric oxygen therapy. So we literally go in and we hit it with oxygen because they can't deal with it very well. Right. Now tetanus, we're going to talk about tetanus and some other things. So I want you guys to know the methodology of how this works. So Clostridium tetani, common resident of soil and GI tracts of animals. All right, so how would we give you a case study on somebody with tetanus, or what would be a key thing that they would put in a case study or question so you know that we're really looking for tetanus?
Puncture wound from a metal object.
Stepping on a nail.
Or a rusty nail or something in the soil, right? So we're going to say, well, we'll look at some neurological symptoms, and then you'll have to see like the person was in the soil or they had a puncture wound, or punctured the skin and got this spore former deep into the tissue, right? Causes tetanus or lockjaw or neuromuscular disease. All right, so what happens with tetanus? All right, so their muscles lock and they can't they can't contract, and we can use this for Botox, right? We're using the toxin for that. Most commonly among geriatric patients or IV drug users, neonates in developing countries also, because they're when they cut the umbilical cord and things, it might not be under complete sterile conditions. Dr. Rowe could tell you a story where somebody some guy chewed the umbilical cord off of the baby.
What the hell?
Very slightly better.
So, yeah. So spores usually enter through accidental puncture wounds, burns, umbilical stumps, frostbite, or crushed body parts. And so some of them are getting deep in the tissue. Anaerobic environment is required for vegetative cells to grow and release toxins. So they have to be in an area where there's not a lot of oxygen. Oxygen will kill it. So technospasms. Neurotoxin causes paralysis by binding to the motor nerve endings. All right, binds to the motor nerve endings here, blocking the release of neurotransmitter for muscular contraction inhibition. So please realize if you're contracting your biceps, remember that your triceps are also contracting slightly so that muscle doesn't contract at 100%. Death most often due to paralysis of respiratory muscles. So if this gets really bad, what's going to happen is your diaphragm is going to be paralyzed, and now you have no way of breathing unless we put you in some kind of iron lung. Right? We don't have time to go over how breathing works, but remember, if your diaphragm goes down, you increase the area in your thoracic cage, you diminish the pressure, the air pressure is higher, forces air in. When the diaphragm releases, it makes the thoracic area in your lungs, all right, makes it smaller, the pressure goes up, you force the air out. That's how it works. Remember that, Robert? No?
Yes. Yeah, sorry, I'm on mute.
Bringing that diaphragm, that little thing out and showing you guys?
Yep.
So you don't need Kevin from like.
So here's neonatal tetanus. Right, so this baby has it. Now that baby literally is in extension. Right? So, at this point, we can't really get rid of the toxins that are there, but please realize we can give them antitoxins to bind up those toxins so we can't cause any further damage. Right? Okay, so treatment is aimed at deterring degree of toxicity and injection and maintaining homeostasis. So once again, the toxins are there. Your body really can't bind them up, especially in the neonates. So we're going to give them antitoxins to bind up any receptors, right, so they can't attach to anything else. antitoxic therapy with human tetanus immunoglobulin, activates circulating toxin, but does not counteract that's what's already around. We hit it with penicillin, tetracycline. Vaccinations available. Booster needed every 10 years. And does anybody remember the shot we talked about?
Well, that's what she's
The
saying.
Tdap.
Oh,
Yeah.
yeah.
That she wants to pull all of his previous MRIs and compare.
All right, I'm gonna restart. If you guys, I don't have any other
She
choice.
just had one. Well,
Um,
the doc-
I'm gonna stop sharing and restart the video right here at 19. Sorry, guys, because we're gonna run out halfway through here.